Welcome to Nucleic Acids Part 3. In this part of the series, we will look at RNA and how the primary structure of proteins is synthesized. Let's get started. All right, so we remember that DNA has the genes that hold the instructions for how to synthesize the proteins, right? But the actual synthesis occurs with RNA and ribosomes. So it turns out that there are 3 major forms, and they each have different jobs. Their names are very helpful though, right? So the rRNA is described as the ribosomal RNA. And these tend to be larger. They typically have 100 to 1,000 nucleotides. And then the mRNA is the messenger RNA. And then the last form is the transfer RNA. And it tends to be the smaller one with only typically 70 to 100 nucleotides. Okay, so now remember, right, that the nitrogenous bases, so for RNA, we have the adenine, the cytosine, and the guanine, but remember that uracil, right, replaces thiamine. Okay, now, so what's the same and what's different? So we've done a lot to learn about DNA. Let's use that to compare and contrast with RNA. So the first difference is the uracil replaces the thiamine. The second is that RNA is a single-stranded nucleic acid versus DNA with the double helix. And then remember from our introduction, right, ribosomes are the protein-making factories of the cell. And so in this tutorial, we're going to focus on 2 main processes. We're going to look at transcription. This is where the instructions from a section of the DNA are copied to the messenger RNA. And then we'll look at translation, where transfer RNA delivers amino acids to the ribosome for the protein synthesis. All right, so now if we look at the schematic, transcription, right, transcription is right here. It's getting the message, right, the instructions to be activated. So from the DNA to the messenger RNA. And then translation will be where the actual protein synthesis occurs. All right, so there, and then if we can take this understanding to the cell, right, so the DNA is in the nucleus of the cell, and then the transcription, the DNA shares the information with the messenger RNA, right? The DNA is trapped inside the cell, but the messenger RNA can come out, and then it can give the directions to the transfer RNA and the ribosome, and then we get the protein synthesized. And so these little balls are representing the individual amino acids. And ribosomes have a very unique shape. You can see they have like the 2 lobes there to help you recognize them. Okay, so let's look at transcription, and then we'll move on to translation. So, right, so remember that we have a segment of the DNA unraveling to expose the nucleotide sequence, right? There's the gene. Right, the nucleotide sequence is the gene that gives the directions for how to synthesize a protein. Alrighty. So then the RNA polymerase, right, so we recognize that as another enzyme, right, can build a complementary nucleotide sequence of messenger RNA. Right, the DNA folds back up. It's shared the information it needed to share. And now the messenger RNA can be exported out of the nucleus and can travel to the ribosomes in the cytoplasm, right? So remember, gene expression is a gene's DNA is used as a template for the synthesis of a specific protein. So gene expression begins with the transcription. So DNA must be unraveled to expose the nucleotide sequence, and then the copying begins at the nucleotides. And so there are codons, there are patterns of nucleotides that signal where a sequence starts and stops. All right, so let's look here. All right, so the whole idea of the template strand of DNA is really important. All right, so let's look down here at our diagram, and here we see the template strand of DNA. All right, so it has unraveled, and then the RNA polymerase comes in to create the complementary strand of RNA, which, is right there. Okay, so there's our complementary strand of messenger RNA comes, right? And so we could think of this part right here, right? This is where we have the gene with instructions to produce a protein. Alrighty. Okay. So one thing to keep in mind, right? So the start codons, that's right. So that's a sequence of nucleotides to signal the beginning of a gene. Or the beginning of the instructions. Alrighty. Okay, so let's think about what we've learned so far about the nucleotides. Alright, so on our template strand right here, oops, I wanted to use green. Alright, so if we have our template, right, so our template is from the DNA, and if it has a sequence, G, A, T, C, A, T, alright, what would be the complement? Right, so the complement will be on the messenger RNA, right? So this is where we have to think about what's the same and what's different about the nitrogenous bases. So for the G, everything's the same. Cytosine matches guanine, but then, right on RNA, there is no thymine, so we would use a U for uracil, adenine, guanine, uracil, adenine. So you want to be able to match the bases up between the template from the DNA and the complement on the messenger RNA. Okay, so that's pretty much all we need to know about transcription for our class. There's a lot more to learn, but that's enough for us. And now we'll go on. So once the messenger RNA has the instructions, how do we actually synthesize a protein? Well, we need to look at translation. Okay, so what happens here, right, is the ribosomes then will take the information from the messenger RNA and read it, and then they'll build a polypeptide by linking the amino acids, right? The primary structure of proteins. Alrighty, so it's interesting to note that we saw this before, the start codons. So what is it about these codons? Well, every group of 3 nucleotides on the messenger RNA is known as a codon because it codes for one of the 20 amino acids. So if we think about, here is our DNA, and then we have the nucleotide sequence, and so we can see here, right? So these three nucleotides code, right, for one amino acid. And then the next three nucleotides code for the second amino acid, and then so on. And then the third for a third amino acid. So this sequence of nucleotides describes a particular amino acid. It's the code for it. However, there are 64 possible codons, and there's only 20 amino acids. And this is really good because when we talked about replication and things, we saw that there is a very small percentage of errors. And so by able to have more than one codon for each amino acid, this helps to minimize effects from mutations or mistakes. So here we have, there's the genetic code. It's the key for the codons and their amino acids. So here we see the first letter, and then here's the second letter, and then you can see how pretty much each amino acid has several different codons to represent it. For example, leucine, there are 4 different ways that we could code, for leucine. So the main point here is the multiple codons, right, help to minimize effects of genetic mutations. Okay, that all fits, great. Okay, now this chart we're going to come back to, but we need to go a little bit further into the story. And the main thing to think about is when you hear the word codon, you're always thinking about messenger RNA, right? Okay, so now let's bring this all together to see how we synthesize proteins. Okay, so here's our diagram down at the bottom. So this is happening, and we'll have another diagram on the next page, right? So codon, think about the messenger RNA, and we'll be thinking always in terms of 3 nucleotides at a time. All right, so then this cloverleaf structure, this represents the transfer RNA. All right, so the transfer RNA is very interesting. So here we have the anticodon. So whenever you see anticodon, think about the transfer RNA. When you think about the codon, you're thinking about the messenger RNA. All right, and then this is the cool part, right? So this codon is giving the code for this amino acid. All right, so the transfer RNA knows, right? So only the transfer RNA that has the correct anticodon can come in to interact with the codon of the messenger RNA so that the transfer RNA brings the correct amino acid. Alrighty, so let's go back up to the notes a little bit and see what's going on here. Alright, so we pretty much just explained that, right? So the transfer RNA matches the codon on the messenger RNA with the corresponding amino acid. The clover-shaped transfer RNA has 2 important regions, right? The anticodon loop, so that's right here. Okay, so the anticodon loop, right? That's where it's the complementary. Well, it's complementary to the messenger RNA codon, right? And remember to focus that it takes only 3 amino acids, okay? And then the 3' end is what is linked, right, to the amino acid. Okay, so that's about all we need to say there. Let's make sure, right, so the CG, or let's see, we have GCC, okay. So now let's go back to the previous page and look, right. If we look at the codons, so we're looking for GCC. Oops, yeah, so sure enough, right, there's GCC right there, right? So that's the code for alanine. So right, remember the GCC is on the mRNA and the alanine is on the transfer RNA. Oops, you can't see what I'm writing. Sorry. Okay, right? So the GCC, that was the codon that tells the transfer RNA to bring in the alanine. All right, so that's right there. So there's our codon and our anticodon and our amino acid. Alrighty. Okay, so now let's look at how this all fits together to synthesize proteins. Building a protein. Alrighty. So now we'll even have the ribosome structure there. Alrighty. So let's look at that. Okay, so at the ribosome, and messenger RNA codon is read, and a matching transfer RNA molecule arrives with the corresponding amino acid. So we saw that on the previous slide. The base pairs H bond temporarily between the anticodon of the transfer RNA and the codon of the messenger RNA. The next transfer RNA arrives with its amino acid, and the transfer RNA base pairs up, right, all through H bonding between the adjacent codons. We get an amide bond forming between the adjacent amino acids on the two transfer RNAs. So let's see if we can look at that part first before we get too far. All right. So it's a little dark, but I think, right? So here's the messenger RNA, right? So that would be our codon, right, to specify. And then we see, right? So here's the transfer RNA, and it has its corresponding amino acid. And now the second, so then we have our second code, and here's the second molecule of transfer RNA bringing in a second amino acid. Everything is held together with H bonds, and now we see that these two amino acids, right, are going to, right, an amide bond forms between them to link glycine to isoleucine. At this point now, the glycine transfer RNA, yeah, it can leave, and we see the isoleucines held in space here. And so now we could bring in a third amino acid, and the process just repeats over and over, and then it's like putting beads on a necklace. We bring together the amino acids to create the polypeptide. So, and that's just summarized for you here, right? The first transfer RNA is released, and then an additional transfer RNA can come in, and this process keeps going until we hit a stop codon, right? And at this point, we're still calling it a polypeptide because the secondary, tertiary, and possibly the quaternary structure has to manifest before we call it a protein, right? Because a protein has biological function, and a polypeptide is simply a polymer of amino acids. All right, so if you look back at your chart, you'll see for this first, if you look back at our codon chart, you'll see that GGG corresponds to the amino acid glycine. And if we look, AUC is isoleucine. Okay, so now let's have one last page to wrap up and practice our understanding. Make sure that we have this understood before I set you free on homework. Let's look at this process one last time. So we'll use the genetic code table at the bottom of the page to help us identify the amino acid attached to the RNA. All right, so if we look here, right, so where are we going to look? Well, right, this is our codon. This is where we need to look, right, because this is the codon table. Since we're here, right, remember this would be called the anticodon, right? That has to do with the transfer RNA. The codon's always the messenger RNA. And then notice, right, this codon links to this amino acid. So the question is, what amino acid is it? Where do we look? We look at the codon. So we would look at GCC. All right, so we look at our table and we go G and C. So if we look GCC, there it is, voila, right? So alanine. So we can now recognize that this is alanine, is the amino acid that will be transferred with this tRNA. Okay, so that wraps up our tutorial on ribonucleic acids and their role in protein synthesis, right? The transcription and the translation are two key processes. You want to be able to find the complements of the nitrogenous bases and be able to interpret the codon table. So please take some time now to reinforce your understanding with a few homework problems.